Well, hi, everybody. Thank you so much for having me join you at your Women in Science and Engineering Technology Symposium. It's a real honor to be here. I would rather actually be there. I have visited Singapore and I quite love the place. So I'm sorry I couldn't join you in person, but perhaps another time. Sorry, I'm just checking my clicker here. Be right back. Okay. So today I'm going to talk about several topics. I was given a somewhat bold set of criteria for my talk and told to talk about both my research on microbiology as well as research on people. So, and that's women in science research. So I decided to do three topics, and that's first, the study of microbial communities and what my lab does to understand communities and how they work. The second is microbes in the global human community. So that's a hybrid of microbes and humans. And then the third is studying human communities. So I will try to keep on time, but cover all three topics. So let's start with microbial communities. One of the challenges that has become clear over the last 20 to 30 years is that microbiology is far more complex than we ever thought. It turns out, underestimated for about 100 years the number of species in the world because we didn't realize that most microbes don't grow under the culture conditions that we use. And so it's an example of this complexity is the human microbiome, which has 100 to probably closer to 1,000 times more genetic variation than the human genome. And we all know the human genome is enormously complex. The soil microbiome is 10 to 100 times even more complex than the human microbiome. So all the bacteria, fungi, organisms in the soil create one of, if not the most complex communities in the world. And that's the world that I want to study and understand is the soil microbiome. One of the problems with microbiology today is that we realize now that there are all these microbes that influence these important life events from the global nutrient cycles. Nitrogen and oxygen and carbon are all dependent upon microbial activity to our own health. There's evidence that illnesses as diverse as colon cancer and depression are managed by our microbiomes. So of course we want to be able to modify those microbiomes. But the problem is that we lack the governing principles. We've never really laid down the basis that would allow us to predict what would make a community stable or what would make it vulnerable to invasion. And so my lab has answered this problem by developing a very simple model system in which we can do a very thorough genetic and chemical analysis of the community. And then coupled with that, I think it's critical that microbial ecology be based on quantitative models that validate findings from one community to another and see, do the same models actually hold true across microbial communities? So we looked for a system that was simple, but still relevant to a natural system. And that's what we named Thor. The answer to that, we named Thor. So Thor starts with Bacillus cereus, which is an organism from the soil and from plant roots that we've studied for many years. We made an odd observation many years ago that you can pick colonies from a plate, what you see on the left, cultured from soil bacteria or root-associated bacteria. You can colony purify them, shown in the second panel, which gives you supposedly a pure culture of just Bacillus cereus by picking the right colony morphologies. But when you take those supposedly pure cultures and put them at 4 degrees, after about 2 to 3 weeks, they start showing these orange or yellowy kind of blebs coming off of the main Bacillus cereus growth. And then on the far right, you can see what happens when you try to culture these organisms away from Bacillus cereus. Typically, but not always, you get these yellow kind of slimy organisms. And it turns out that those are largely from the phylum Bacteroidetes. So about 83% of these organisms that co-isolate with bacillus from roots are Bacteroidetes. And then the others are kind of a mixture of other phyla. We call these the hitchhikers because they come into the lab as co-isolates with Bacillus cereus. And that's where Thor gets its name. The hitchhikers of the rhizosphere is what Thor stands for. So we found an interesting phenomenon. About a decade ago, we found that one of these hitchhikers, or a group of them that are members of the Bacteroidetes, are grossly stimulated in their growth and culture by Bacillus cereus. And it turns out that only the organisms in the Bacteroidetes are stimulated by Bacillus cereus. And you can see the difference in growth at inoculation, the population at 3 days, the population at 5 days. And all of the columns that you see here with the red bars are Bacteroidetes. And on the right, I've grayed out the other kinds of organisms that come along as co-isolates, because those are not affected by Bacillus cereus growth, by Bacillus cereus in their growth. And it turns out that the Bacteroidetes are specifically stimulated in growth by being provided peptidoglycan, which is the cell wall fragments that Bacillus cereus sloughs off when it divides into two cells. And so on the upper right here, you see fragments of peptidoglycan, the structure. And we find that growth is directly proportional to the concentration of peptidoglycan in the media. So we began to study Flavobacterium, which is one of these Bacteroidetes, in more detail. And we discovered very recently a very odd phenomenon that we call a walking biofilm. This seems to be a new mode of bacterial movement, and we call the structures that are formed zorbs, and they have a very, very interesting etiology. So the first thing that happens is Flavobacterium will attach to a surface. Flavobacterium is a gliding bacterium, so it has a very unusual mode of movement, motility on a surface to begin with. But then when we put the Flavobacterium johnsonii on this surface, we find that after a few hours, they begin to form these globular structures, so aggregates of bacteria that you see in the middle panel, and then eventually get much larger, and we call these the zorbs. They look like and act like biofilms. They appear to be held together by polysaccharides, which is typical of biofilms, and yet they move, they just slide across the surface. And so this is one of the few examples we've ever seen of a moving biofilm. And that's what we call it, the walking biofilm, because it's a very unusual phenomenon. Biofilms are usually stationary. That's kind of what we think of biofilms as doing, is anchoring bacteria to a surface. And eventually these zorbs or aggregates will dissociate and go back to single cells over a long period of time. So I wanted to show you an example of what these zorbs look like and what happens when you have Flavobacterium and Bacillus cereus together. So you'll see Flavobacterium in an orangey red color, so it's fluorescently labeled, and you can see the zorbs beginning to form. That's all Flavobacterium. And as the zorbs get very structured, you can begin to see Bacillus cereus, which is in green, inside the zorb structure. So this is a very odd phenomenon that has never been seen before. Sorry. Of bacteria forming these globular or spherical structures, and that's the Flavobacterium, encapsulating another bacterium called the Bacillus cereus and the internal part, and being able to move as a co-structure, structured by both of them. And here, just so you can see it one more time, you can see Bacillus cereus, the green, inside the zorb, and it looks like it penetrates the zorb from the outside. So the zorb may form, and then Bacillus cereus enters it, and somehow it's like a reverse hitchhiking, whereas Flavobacterium hitchhiked into the lab from the field within cultures of Bacillus cereus. In this case, it looks like Bacillus cereus is hitchhiking in these mobile structures, the walking biofilms. We began to look for a third member for this community. Remember, we're trying to develop a model community that would allow us to dissect the microbes in it in a very detailed way. And it's not a community unless it has more than 2 members. So we wanted one more. And so we found that among the other hitchhikers that were not members of the Bacteroidetes, like Flavobacterium, there were many pseudomonads. And interestingly, one of them, when we tested it against all the other co-isolates, we found that it inhibits the growth only of Bacteroidetes. So if you look at the bars on the right, this is with the isolate is cultured alone in blue, or with this Pseudomonas coriensis. And you can see on the right, the blue and yellow and orange bars are about the same. There's no significant inhibition. But if you look at the blue bars on the left, those are all Bacteroidetes. And the reason you can't see the orange bars is there is absolutely no growth that we could measure among all those different Bacteroidetes strains when they're co-cultured with Pseudomonas. So this was the first known antibiotic that was specific for the Bacteroidetes phylum, and that was really fascinating to us. And so we thought, well, this would make an excellent additional member for our community. So if you look at these structures, these are the antibiotic we discovered that the Pseudomonas produces. There are several related structures, but the one we think is the most active is coriensin B, which is in the box in the upper right. Very simple structure, actually closely related to conine and conicine, which are the toxic compounds in the hemlock tree, but had never been seen, nothing like them had ever been seen in bacteria. So we now have a microbial community that has antibiotic interactions and nutritional interactions. And we began to look at the physical behaviors on plates at a macroscopic and then at a microscopic level on plants. So on the left, you can see the behavior of Bacillus cereus when it's cultured alone in the first 2 windows. In the middle, you see Bacillus cereus when it's cultured with Flavobacterium johnsonii. And you can see that it starts forming this sort of dendritic growths out from the colony. And similarly, when it's cultured with Pseudomonas coriensis, the third member of Thor, again, we see these dendritic growths. On the right, we see some of the interactions that occur on soybean roots. So this is where several of our organisms were originally isolated, and all of them seem to grow on soybean roots. So in this case, we're looking at Bacillus. So the first column are roots, the root tip of plants that have been inoculated with Bacillus. And the Bacillus forms this interesting structure only at the root tip. So you can see it kind of decorating the end of the root. But in the second column, we see Bacillus with Flavobacterium, looks fine. In fact, the ring structure in that middle one looks even brighter. But in the third column, you see Bacillus plus the Pseudomonas coriensis. And we completely lost this structured organization of Bacillus cells around the root tip. And the final panel, you see that when we add Flavobacterium back, a little bit of that structure at the root tip begins to come back, but it's nowhere near as defined and distinct as the Bacillus alone. So we don't know the basis of this. This is under investigation, but these are the kinds of interactions that this model community exerts. On the bottom panel here, we see an example of the biofilm that the organisms form together. And interestingly, they seem to produce more biofilm when they're together than any of them individually or in pairs. The blue is the Pseudomonas, and so you can see that is the dominant organism in the biofilm. The bluish green cells are bacillus and the pink ones are flavobacterium. So it's particularly interesting that even though flavobacterium and bacillus are less than 10% of the biofilm, they have a fairly dramatic effect on the amount of biofilm that's formed. Next slide, please. So this is just a summary of the interactions that we've discovered to date among the members of Thor. So at the top, you see Bacillus cereus, and then Flavobacterium on the left and Pseudomonas on the right. And so we know that there's a nutritional interaction between Bacillus and Flavobacterium. Flavobacterium eats the cell wall that Bacillus sloughs off. Of course, Flavobacterium is also a hitchhiker on the Bacillus cereus cultures coming into the lab, but then Bacillus cereus seems to be a hitchhiker in the sorbs formed by Flavobacterium. If we look at the Bacillus and Pseudomonas interaction, we find that, again, Pseudomonas comes into the lab with a Bacillus culture as a hitchhiker. But then an antibiotic produced by Bacillus seems to be involved in that enhanced biofilm effect that I told you about, and I won't have time to go into that. Another thing that we found is that if we skip down to the interaction of Flavobacterium and Pseudomonas, we find that Pseudomonas produces an antibiotic called coriansin. I showed you coriansin that inhibits growth of Flavobacterium, and yet it doesn't seem to inhibit Flavobacterium in the presence of Bacillus. So it looks like Bacillus inhibits production of coriansin. Another interesting anomaly is that Pseudomonas doesn't produce very much coriansin by itself, but instead it responds to the presence of Flavobacterium. So in kind of a warped interaction, Flavobacterium is inducing Pseudomonas to produce the very chemical that will kill Flavobacterium. But Bacillus is, of course, smart enough to protect Flavobacterium from that antibiotic. So these are the interactions we know of to date. There's a lot more to learn about this community. We have all the genetic tools and the genomes are sequenced. We have the ability to look at the full transcriptome of the whole community. And we are looking at the metabolome of the community because we've already found so many interesting small molecules that have either antibiotic or signaling activity. So we think that in a few years, we'll be able to describe this model community in exquisite detail and begin to determine whether the factors that regulate its behavior are important in other communities as well. So I'd like to turn to my second topic today, which is the interaction of microbes in a global human community. And that's the Tiny Earth Project. So this started as an educational initiative that was intended to teach students biology in college, but also to address the very serious antibiotic crisis that we're facing worldwide. And that's due to the fact that antibiotics and that bacterial pathogens have developed enormous antibiotic resistance. Some of the pathogens are resistant to all of the known antibiotics used against them. And so we've lost even our last line of defense for some pathogens. And that's coupled with the lack of a pipeline of new antibiotics. So the pharmaceutical industry worldwide pretty much withdrew from antibiotic study in the 1980s and 90s. And so the pipeline is running dry just at the time that we're reaching the end of the line on compounds that can treat so many human diseases where the pathogen is getting resistant. So we decided to develop this course that involves undergraduate students worldwide in, first of all, taking soil samples from the environment where they live. They then culture the bacteria from soil, and then they begin to screen those bacteria for inhibition of human pathogens, and they test them against several different pathogens. They then will find the ones that inhibit the growth of the pathogens. There we go. Sorry. And you can see these zones of inhibition around the isolates that students have found in the soil. That's an indicator that those isolates are inhibiting the growth of the pathogen that is spread on the plate, which is the first indication that we have an antibiotic. Then at Madison, UW-Madison, where I work, we then do the chemistry. If the students don't complete the characterization of the molecules active in these isolates, then they can send them to us. We will then do a chemical analysis and try to determine what the compound is responsible for this activity. So this network of students has grown just enormously. We have adopted the course at about 300 institutions worldwide. This covers 42 of the 50 US states and 17, actually we're well above 17 countries. I think this is, we're up in the 20s, probably about 27 countries now. And more than 10,000 students take the course per year. We offer training for the instructors, and that training goes on twice or 3 times a year. Even during COVID, we've been able to offer training virtually. And so we're constantly increasing the number of students taking this course. So this is a way of using a worldwide community, a global form of student sourcing, as we call it, antibiotics from the soil. And we're very hopeful that this will yield many useful compounds for human health. It also teaches a lot of biology to these students over the course of the semester. So this is just an example of some of the sites, certainly not all of them. We couldn't fit all 300 on the map, but some of the sites that we work with Tiny Earth collaborators in. And I just note that the blue one on the far right is not Singapore. It is in Malaysia, and we do not have a Singapore site, and we'd be delighted to have one. So if somebody here would like to join the Tiny Earth Network and discover antibiotics with us and teach biology, we would be thrilled to have a Singapore collaborator. So finally, I'd like to turn to my work on the study of human communities and the interactions of people within communities, particularly of scientists. We know that the diversity of people is directly associated with the creativity of groups. So putting people together in heterogeneous groups leads to better solutions to problems, more creative solutions, and better and more defensible solutions or decisions. But the problem with this is that at the same time that we should be valuing diversity, there's a lot of evidence that many of us, if not all, have certain prejudices against certain groups being more or less able to do science. So just at a time when I think we are desperate for solutions and great science to be done, we need the most productive and creative teams of scientists we can get. We also have this problem of keeping diverse people who perhaps don't look like us or haven't come from the same place as us or something else or have a different gender out of our communities of scientists. And that has resulted in a homogeneity that's simply not healthy. There's enormous amounts of evidence of bias that people carry with them. And I'd like to talk a little bit about how that work was established. It was shown through randomized controlled studies for about 50 years. People did these studies, still do, where they would give someone a candidate to review for either a job or acceptance into a program or a promotion. And they would set it up so that in a randomized system, each person, each evaluator evaluated one candidate, and they were randomly assigned either a man's name or a woman's name, or in some cases, there was an indication that they were of different races or ethnicities. And so the only difference was either the gender in most of the cases or perhaps ethnicity. And what was found was that there was in every one of these studies a significant effect of the gender, or in the case of race, the race of the person being evaluated. And that meant that women and non-white people, people of color, consistently were discriminated against in these studies. Now, people were not intentionally discriminating, and if you ask the people who were doing the evaluations whether they were biased, they would say to a person that they were not biased and they were working absolutely not to have prejudices. These are what we call unconscious or implicit biases that people carry that they don't realize they have, but they unintentionally apply to their evaluation of other people or their work. So this field went on for many decades and found two things consistently. One, what I just said, that there was a significant effect of the gender or the race of the person being evaluated. So you'd have the same record, but if it had a woman's name or a man's name on it, they would get a different result. And just as interesting, there was never in any of these studies a significant effect of the gender or race of the person doing the evaluation. So what that says is that women discriminate and carry these intrinsic biases just like men, and people of color carry similar biases to people of white people. So that's a really interesting phenomenon because it says that we all have somehow absorbed or been inculcated with these implicit biases, and just having a woman making the decision does not necessarily advantage a woman. Next slide, please. So I talked for many years about this research and talked about why we have to worry about such implicit biases in science, because we were actively discriminating against the very diversity that would give us the most dynamic and exciting and interesting scientific community. And in many cases, just about every seminar I gave on this, there would be resistance to the idea of bias. And there were several lines of resistance, but the one that I couldn't answer was the statement that we're trained to be objective, so these bias studies don't apply to us. People would say, we're scientists don't do this. But that doesn't quite make sense because this is an unconscious process. It's not our conscious cognitive processes, which is exactly where we try to be objective in science. And the second thing is that we're actually not so objective in science. If we were objective, why would we do blind studies where the investigator doesn't know the nature of the samples or the people? So clearly we don't entirely trust our objectivity in other areas. So why would we think that we wouldn't apply these biases that everyone else seems to have to our selection of people or our evaluation of their work? So I kept hearing this, and it seemed like people were going to continue to dismiss the evidence. And instead of getting down to thinking about, well, what can we do about bias? How can we reduce the impact of bias? They were dwelling on instead, well, we don't do this because we're not, we're scientists and we're trained to be objective. Next slide, please. So when I was at Yale, I had a postdoc, Corinne Moss-Racusin. And Corinne and I decided, in response to many of these reactions that we had from colleagues that, oh, we're not biased because we're scientists, we decided the only answer was to do the experiment that had been done many, many times in the field of social psychology with other kinds of people, that it was just critical that we do it with scientists, or the scientists would never believe it. So we developed a study of biologists, chemists, and physicists at research universities in the United States. We sent the participants a description of a student and randomly assigned the name either Jennifer or John. Back here, sorry. We randomly assigned the same, so remember, one description, and we randomly assigned the description, either the name Jennifer or John. And we asked, this was a description of a student who was about to graduate from college. And we asked the faculty that we were using as our evaluators, would you hire this student as your lab manager? How competent do you think the student is? Would you provide mentoring for the student? And what salary, if you did hire them, what salary would you offer? And so the first question is, would Jennifer or John be hired? And so here you see the result with, on a 5-point scale, there's a pretty substantial difference, well over a whole point difference in the probability of John being hired and favored over Jennifer. So that was the first thing, which was not a surprise to any of the social psychologists because they had shown this so many times with many other kinds of people. But the scientists all found this, natural scientists found this very shocking because they had believed that they were immune to these kinds of biases because of their training in objectivity. We asked, would you give mentoring to this student? Again, John had a substantial advantage over Jennifer in the faculty intending to mentor. And when we asked what the salary would be, there was a pretty substantial difference, about a 15% difference in the salary that would be offered to Jennifer versus John. So remember, these are identical applications. There are no differences in the qualifications of this student. Simply what name is assigned to the student was the difference. And just like all the other studies in this field, there was essentially no difference between male and female faculty in their evaluation. So on the left, you can see the evaluation of John, and the female and male faculty were essentially the same. On the right, you see the evaluation of Jennifer, and again, the male and female faculty evaluated her about the same as each other, but obviously less positively than John. So this verified that, in fact, we do have a tremendous bias issue. And the question now becomes, as scientists, hopefully we want to be objective. We don't want bias creeping into our evaluations of people and deciding who we're going to hire, who we're going to promote, or whose grants we're going to fund, or whose papers we're going to accept. And so the question then is, how do we manage that bias? We can't get rid of these biases. These are intrinsic to people. They're developed at a very, very early age. There's evidence that the biases are adopted before kids are 2 years old. So we're not going to be changing the presence of bias in the world, but we have to figure out ways to manage it if we're going to give all people a fair chance at jobs, acceptance to school, graduate school, undergraduate, and funding their work and publishing their papers. So we've got to be able to figure out how to manage this, or we have an unfair, non-meritorious system, and that's only to our detriment because it will decrease the diversity and heterogeneity of the scientific community, because we will continue to favor white men throughout all of these activities in science. So there was an interesting hint about what we can do to manage biases in one study that was done, where they asked not only who would you hire, but then they asked why. And they found the typical result that they'd be, the participants would be more likely to hire whichever application had the man's name on it. But in this case, they had two slightly different applications. And when they asked people why they hired one person or the other, they found that the justification was always given by whatever characteristic was stronger in the male's application. So if the man had more education, then they would say, oh my, you need education, a lot of education for this job. And if the man's application showed more experience, they'd say, oh yeah, you gotta have experience for this job. That's the most important characteristic. So this is what we call an after the fact justification. And so we make these biased choices, but we don't want to say, oh yeah, I picked that application because it was a man. That none of us want to be doing that. We don't believe that's what we're doing. But in fact, we pick on elements of an application and choose to focus on them and value them based on the gender of the person who has that characteristic. So they went back and they did this experiment again. And they had a new set of participants identify their hiring criteria before they started reviewing the applications. And they found that when they did that, there was very little bias. They abolished the bias almost completely. And that suggests that when we set up criteria and we try to make this an objective process rather than going with a sort of gut level, I know it when I see it kind of choice, then we begin to make decisions based on qualifications and not based on gender. So this gives us a clue for what we can be doing in, for example, reviewing people for jobs or grants or promotion or acceptance to graduate school. We should be setting up our criteria ahead of time and making sure that we are evaluating everybody according to the same criteria. Some of the other examples of activities, practices, and interventions that have been used to reduce bias are one where they had people play a board game that showed the disadvantages of women in a particular tenure system, and then evaluate men and women. And it turned out that just going through the process of playing that game reduced people's biases. Several studies have now done blind reviews. One journal in ecology actually for 6 months took the authors' names off the papers, and they found that there was a 30% increase in the acceptance of papers who were senior authored by women during that period of time. So that indicated that not knowing the gender really did make a difference in the evaluation. There have been several studies now of evidence-based training where you teach scientists about the kind of data that I described to you in the last several slides, where you actually go through the studies. Scientists are swayed by data. So let's use the data and teach them what the data show in terms of bias. And some of those programs have been shown to increase the hiring of women and change attitudes about the importance of bias in the process. Because if we don't admit there's bias, it's very hard to combat it. One of the things that has always interested me is the effect of imagery and art on our attitudes. And you may have noticed that many of my slides have pictures of women scientists on them. And that comes from some of the studies that show that exposing people to examples of strong counter stereotypes, we might call them. So people who don't look like the stereotype scientist or whatever the position of interest might be, break down the bias. So people begin to get used to seeing powerful women in science. They will then evaluate women in the future with a little bit less bias. One of the problems with these kinds of interventions is that their effect doesn't seem to last that long. They do have a strong effect, but in some cases, the effects only last 24 hours, which means that you have to continually show people these images or present them with fictional people who can demonstrate the power of women or minorities or people of different abilities or disabilities in science, and this can begin to reduce their biases. And so next slide, please. So we developed, after we did the bias study with Jennifer and John, we created a set of videos, which you can find at this website. It's called the VIDS Project. And the idea is to use videos of scenarios of men and women interacting in science to begin to make people aware of the kinds of biases that they bring to the table. And so these are very typical interactions, a seminar given by a male and a female student who are treated quite differently, a faculty meeting where there's discussion of a candidate that is being considered for a faculty position. So these are very familiar situations to people in academic science, and they're just 5 minutes each. And we showed them to participants in our study, and then looked to see what the effect on those participants' attitudes and behaviors were, and found that in fact being exposed to these videos did change their attitudes about women in science and made them aware of the biases that continued to plague us. So what can each of us do? Clearly, these biases muddy our thinking and distort our decisions about who to hire and reward. And they're not related to merit, they're related to characteristics that have nothing to do with people's success in science. So the first thing is everything I've been talking about in the last few slides is examine our own biases and point out biases for other people. I think it's critical that we make bias a common part of our discussions. So when we're holding a search committee meeting, for example, we talk about bias and we ask ourselves, how might it be entering the process? How might it be influencing our evaluation of these candidates? And if everyone holds not only themselves, but their colleagues accountable for being as unbiased as possible and rooting out bias and preventing each other from applying it, I think we can make tremendous strides toward having a much more meritocracy in science, and ultimately a much more heterogeneous and diverse community of scientists, and that will lead to better science. So finally, I'd like to thank you for your attention and all the great scientists I've worked with on the projects I talked to you about, and the women faculty that are in my institute, the Wisconsin Institute for Discovery. I have the privilege of working with 6 other really amazing women in science, very diverse fields from statistics to stem cells, but they all bring their own perspective and their own contribution to the community of scientists. And I think it's one of the things that makes our institute as exciting a place as it is and the science as good as it is. So with that, I will stop and thank you very much for the invitation to join you and for your attention.